Have you ever wondered what stories your DNA could tell? What if the history of an entire nation, its migrations, empires, triumphs, and tragedies, was encoded in the blood of its people? From ancient innovators and Sahelian traders to the scars of the slave trade and the dawn of a global diaspora, we unravel the epic 25-part saga of why the DNA of Ghana is one of the most unique and complex on Earth. Deep within the transitional zone where Ghana's dense forests give way to the sprawling savanna, a quiet but profound transformation of human existence unfolded, laying a foundational genetic and cultural stratum that continues to resonate in the DNA of modern Ghanaians. This was the era of the Kintampo Complex, a prehistoric culture that flourished between 2500–1400 BCE, representing one of West Africa's earliest transitions to a sedentary, food-producing way of life. The people of Kintampo were not simply passive recipients of environmental change. They were active innovators, ecological engineers who fundamentally reshaped their relationship with the land, and in doing so, initiated a new trajectory for human society in the region. The archaeological record, unearthed from sites like Kintampo, K6 and Bairimi, reveals a society of remarkable sophistication and resilience. They constructed permanent villages, a radical departure from the nomadic patterns of their hunter-gatherer ancestors. Their homes were not flimsy, temporary shelters but sturdy waterland daub structures, often built upon foundations of packed earth and stone, clear evidence of an intention to remain. This commitment to place fostered the development of complex social structures, the accumulation of generational knowledge, and the birth of ancestral traditions tied to specific landscapes. This sedentarism was underpinned by a revolutionary agricultural package. The Kintampo people were pioneers in the exploitation of the oil palm, a plant that would become a nutritional and economic cornerstone of West African life for millennia. They also cultivated other crops like cowpeas and yams, and herded small livestock, particularly dwarf goats, indicating a balanced and sustainable mixed economy. This agricultural lifestyle exerted a new set of selective pressures on their biology. Genetic adaptations related to digesting a more carbohydrate-rich diet, processing specific plant toxins, and developing immunities to pathogens associated with settled life and animal husbandry likely began to proliferate within this population. The very act of clearing land, cultivating crops, and managing livestock created a new ecological niche, and the Kintampo people's DNA began a long and intricate dance of adaptation with this environment they themselves had helped to create. Beyond subsistence, they nurtured a rich artistic and spiritual life, evidenced by their distinctive material culture. They crafted a characteristic style of pottery, often decorated with intricate comb-like impressions, a shared aesthetic that connected their disparate settlements into a cohesive cultural sphere. Most iconic, however, are the enigmatic terracotta figurines, often cylindrical and dubbed Kintampo cigars, which, while their exact purpose remains debated, unequivocally point to a complex symbolic world. These artifacts, along with polished stone axes and bone harpoons, were not merely tools but expressions of a shared identity that was transmitted across a wide geographical area. The existence of such a widespread and enduring cultural complex implies a network of interaction, trade, and communication linking their villages. This network was a conduit not only for goods and ideas but also for genes. Through intermarriage and small-scale migration between settlements, a regional metapopulation was forged, creating a foundational gene pool characterized by a shared set of adaptations and a common, deep-rooted ancestry. This was the genetic bedrock of the region, established long before the arrival of the great migratory waves that would later define Ghana's ethnic landscape. The Kintampo people were the first to truly domesticate this part of West Africa, and in the process, the environment began to domesticate them, shaping their genetic makeup in ways that would prove foundational. Their legacy is not written in grand monuments or epic texts but is inscribed in the land itself and, more profoundly, in the subtle genetic markers carried by millions of Ghanaians today, a silent testament to the ancient architects who first called this land home. Long before the modern nation of Ghana adopted its name, a powerful and immensely wealthy empire of the same name thrived hundreds of miles to the northwest, in what is now southeastern Mauritania and western Mali. The Ghana Empire, or Wagadu as its Sonic founders called it, reached its zenith between the 9th and 11th centuries, and while it had no direct geographical overlap with the modern state, its collapse triggered a cascade of migrations that would profoundly shape the genetic and cultural landscape of the entire West African region, planting deep ancestral roots that are still traceable in the DNA of many contemporary Ghanaians. The empire's power was built on its strategic control of the Trans-Saharan trade routes, acting as the critical intermediary between the salt mines of the Sahara and the goldfields of Bambuk and Beret. This commerce created a cosmopolitan society where Sonic people mingled with Amazigh and Arab traders from the north, and with various Monde-speaking groups from the south. This imperial heartland was a crucible of genetic exchange, a vibrant melting pot that concentrated diverse lineages from across the Sahel and North Africa. However, the empire's dominance was not to last. A combination of factors, including internal strife, the disruptive invasions of the Almoravids in the 11th century, and increasing environmental pressures from desertification, led to its gradual decline and eventual fragmentation by the 13th century. This collapse created a power vacuum and widespread instability, forcing countless communities to seek new homes and new opportunities. It was this period of dispersal, the great unraveling of the old imperial order, that set in motion the population movements that would directly impact the future territory of Ghana. Groups of Monde-speaking peoples, including ancestors of the Daïla traders, as well as various Proto-Akan groups, began a long and generational southward and eastward migration. They were not moving into an empty void. They encountered pre-existing populations, such as the descendants of the Kintampo culture and various Gur-speaking communities, who had inhabited the northern savanna and forest transitional zones for centuries. The encounter between these migrating groups from the collapsing Sudanic state and the indigenous inhabitants was a pivotal moment of admixture. The newcomers brought with them not only new technologies, such as advanced ironworking and state-building concepts, but also a distinct genetic inheritance forged in the imperial melting pot. This included Y-chromosomal and mitochondrial DNA haplogroups with a more Sahelian and, in some cases, North African character. The oral histories of many of Ghana's most prominent ethnic groups, most notably the Akan, retain strong memories of this northern origin, speaking of a migration from the Great Desert or from the vicinity of a powerful ancient kingdom. These traditions are not mere myths. They are cultural echoes of a real demographic and genetic event. The integration of these migrant lineages with the indigenous gene pool created a new, complex synthesis. This process explains the presence of certain genetic markers in modern Ghanaian populations, particularly in the northern and central regions, that connect them to the broader West African Sahel rather than just the Guinea coast. The legacy of the Ghana Empire in modern Ghana is therefore not one of direct political succession, but of demographic and genetic inheritance. The empire's fall was the catalyst that pushed a wave of humanity southward, carrying with them the genetic echoes of a once great civilization. These echoes became a crucial layer in the palimpsest of Ghanaian DNA, adding a Sudanic and Sahelian signature to the ancient Guinea Coast foundation. The story of the Akan people, who constitute the largest ethnic group in modern Ghana and Côte d'Ivoire, is a saga of migration, state-building, and the profound influence of a unique social structure on the very fabric of their biological inheritance. Their southward movement from the Sahelian regions, likely spurred by the disintegration of the Ghana Empire and the subsequent search for new lands, unfolded over centuries, culminating in their settlement in the rich, gold-bearing forest regions of present-day Ghana. Here, they established a series of powerful states, from the early Bono state to the later Denkyira, Akwamu, and the formidable Ashanti Empire. While their political and military achievements are well documented, it is their matrilineal social organization, the Abujua, that offers the most fascinating insight into the uniqueness of their DNA. For the Akan, identity, inheritance, and succession are not traced through the father's line, but exclusively through the mother's. A child belongs to their mother's clan, and a man's heir is not his own son but his sister's son. This social principle, enshrined in their culture for centuries, has had a direct, measurable, and powerful impact on their genetic landscape, acting as a cultural force that has actively shaped the flow and preservation of specific genetic lineages. Biologically, this system places immense importance on mitochondrial DNA, mtDNA. Unlike nuclear DNA, which is a mix from both parents, mtDNA is passed down almost exclusively from mother to all her children. It is a direct, unbroken line of maternal descent. The Akan social structure has created a near-perfect alignment between this biological reality and their cultural identity. The 8 major Akan matriclans, the Oyoko, Brechuo, Adwana, Asona, Asani, Akwona, Agona, and Asakiri, are, in genetic terms, vast families of distinct maternal lineages. For centuries, the Abujua system has acted as a powerful vessel for preserving the integrity of these ancient mtDNA haplogroups. While men might marry women from other clans and their Y chromosomes would spread across the population, the core maternal lineage of each clan remained anchored, passed faithfully from mother to daughter, generation after generation. This has created a distinct genetic pattern observable today. Studies of Akan populations often reveal a high degree of mtDNA differentiation between different clans, even within the same geographic area. The maternal genetic signature is tightly clustered according to clan affiliation, a direct reflection of the social rules governing identity. In contrast, the Y-chromosomal DNA, Y-DNA, passed from father to son, tends to be more geographically diffuse, reflecting a history of men moving for marriage, trade, or warfare. The Abujua is therefore a form of social DNA that has meticulously curated the biological DNA of the Akan people. It has ensured the survival of ancient maternal lines that might otherwise have been diluted or lost in a patrilineal system. This makes the Akan a remarkable case study for genetic anthropologists, demonstrating how deeply cultural practices can influence population genetics. The uniqueness of the Akan genetic signature is not just a product of who their ancestors were or where they came from, but is inextricably linked to how they have organized their society and defined kinship for centuries. It is a testament to the idea that culture is not just a layer on top of biology, but an active participant in the shaping of the human genome itself. For more than 1,000 years before the first European ships made landfall on its shores, the territory of modern Ghana was intricately connected to a vast and sophisticated network of trade that spanned the African continent. The Trans-Saharan and Sahelian trade routes were the arteries of a vibrant economy, channeling not only goods but also ideas, cultures, and, crucially, genes across immense distances. The northern regions of Ghana, in particular, served as a critical nexus, a genetic crossroads where the forest belt of the Guinea coast met the sprawling, arid landscapes of the Sahel and the Sahara. This constant, centuries-long exchange created a uniquely diverse and complex genetic mosaic, profoundly enriching the region's gene pool long before the start of the Atlantic era. The primary commodities driving this trade were gold, sourced from the Akan forests, and kola nuts, a valuable stimulant, which were exchanged for Saharan salt, a vital necessity in the tropical climate. However, the caravans that plied these routes carried much more. Textiles, beads, leather goods, horses, and books flowed south, while ivory, spices, and enslaved people were transported north. Bustling market towns like Bago, and later Salaga and Kintampo, emerged as cosmopolitan centers of commerce. These were not merely places of transient exchange. They were melting pots where people from vastly different backgrounds lived and worked side by side. Mande-speaking merchants, known as the Daila, were instrumental in this network, establishing diaspora communities far from their homelands in the Mole Empire. Hausa traders from modern-day Nigeria formed their own distinct quarters, zongos, in these trading hubs. Fulani pastoralists moved with their herds along the savannah corridors, and Arab and Amazigh, Berber merchants, representing the northern termini of the routes, ventured south, bringing with them the cultural and genetic influence of the Islamic world. This sustained interaction led to inevitable and continuous gene flow. The merchants, scholars, and artisans who settled in these towns, often for generations, formed relationships and had children with local Ga and Kwa-speaking women. This process was not a single, dramatic migration, but a slow, steady, and persistent infusion of genetic diversity. It introduced a wide array of Y-chromosomal and mitochondrial DNA haplogroups into the local population that were more characteristic of Sahelian, North African, and even Middle Eastern populations. For instance, the presence of the Y-DNA haplogroup EM81, which is most common among Amazigh populations in the Maghreb, or specific mtDNA lineages of Eurasian origin found in northern Ghanaian groups, can be directly attributed to this ancient commercial network. Islam, which spread along these same routes, further facilitated this exchange, creating a shared cultural and legal framework that encouraged connections between disparate peoples. The trade routes created a genetic gradient, with the highest diversity and most significant Sahelian admixture found in the north, gradually decreasing as one moves south toward the coast. This ancient land-based globalization made the northern savannah a crucible of human diversity, a place where different streams of African humanity converged and mingled. It ensured that the Ghanaian gene pool was never isolated, but was instead continuously enriched by connections to the wider continental tapestry, creating a layered genetic history that reflects centuries of commerce, migration, and cultural exchange across the sands of the Sahara. The year 1471 marked a pivotal turning point in the history of the Guinea coast, an event that would initiate a new and unprecedented form of genetic exchange. When Portuguese navigators, driven by a quest for gold and a sea route to the Indies, first landed on the shores of what they would name the Gold Coast, they inaugurated 5 centuries of direct and sustained contact between Europeans and sub-Saharan Africans. This encounter, initially centered around fortified trading posts, began a slow but irreversible process of genetic admixture, introducing European DNA into the coastal West African gene pool for the first time. The Portuguese, followed swiftly by the Dutch, English, Danes, Swedes, and Brandenburgers, did not arrive as colonizers in the modern sense. Their primary objective was trade, and their presence for the first few centuries was largely confined to the formidable stone castles and forts they constructed along the coastline, such as the iconic Elmina and Cape Coast castles. These forts were isolated European islands in a vast African sea, wholly dependent on the goodwill and cooperation of the powerful coastal states, primarily the Fante, Ga, and Ewe. The population within these forts was overwhelmingly male, a mixture of soldiers, merchants, administrators, and artisans. Far from home and with few to no European women present, these men inevitably formed relationships with local African women. These unions ranged from fleeting encounters to long-term, stable partnerships that were often formalized through local customs and were essential for cementing trade alliances and navigating the complex social landscape. The children born of these relationships gave rise to a new and distinct social group, the Euro-African community. Known variously as mulattoes, cabassiers, or by their European-derived family names, these individuals occupied a unique and often influential intermediary space. They were bridges between the two worlds, fluent in both European and local languages, educated in the intricacies of both cultures, and perfectly positioned to act as brokers, traders, and political advisors. Their presence added a new, visible layer of genetic diversity to the coastal societies. Genetically, this process resulted in the direct introduction of European Y-chromosomal haplogroups, such as R1b, common in Western Europe, into the local maternal lineages. Because these unions were almost exclusively between European men and African women, the flow of genes was unidirectional. European Y-DNA entered the African mtDNA pool. Over generations, these European paternal lineages were passed down, creating families and entire lineages with a distinct genetic and cultural heritage. Surnames that are common in Ghana today, such as Van der Puig, de Graaf Johnson, Lutterot, Hesse, and Bannerman, are living testaments to this history, tracing their origins directly back to a European male ancestor who lived and worked on the Gold Coast centuries ago. While the overall percentage of European admixture in the total Ghanaian population remains relatively low, it is significantly concentrated in specific coastal communities and families, particularly among the Fante people who lived in the shadow of the largest and most numerous castles. This initial phase of contact established a pattern of interaction and admixture that would be greatly amplified and tragically distorted by the subsequent horrors of the transatlantic slave trade. But it stands as a distinct historical and genetic event in its own right. It was the beginning of the Atlantic chapter in Ghana's genetic story, a chapter written not by conquest or mass settlement, but through the complex and personal interactions that took place within the stone walls of the coastal trade forts. Along the arid coastal plains east of the Volta River, centered around the bustling modern capital of Accra, lived the Ga-Dangme people, a group whose unique history of migration, settlement, and interaction has created a distinct and complex genetic profile. Their story is one of a journey from the east, a strategic adaptation to a unique lagoon-dotted environment, and a prolonged, intense engagement with multiple European powers, all of which has left an indelible mark on their DNA. Ga-Dangme oral traditions speak of a protracted migration from a place called Same, believed to be in the region of modern-day Nigeria or Benin, possibly linked to the ancient Yoruba city of Ile-Ife. This eastward origin distinguishes them linguistically and culturally from their Akan neighbors to the west. They brought with them the B languages and a patrilineal social structure, where identity and inheritance are traced through the father's line, a stark contrast to the matrilineal system of the Akan. This foundational migratory event means that the Ga-Dangme gene pool shares significant affinities with populations in Togo, Benin, and Nigeria, representing a distinct stream of genetic heritage that flowed into Ghana from the east. Upon arriving in their present homeland, they did not form a single, centralized state, but instead organized themselves into a series of autonomous towns, or mansamai, each centered around a coastal lagoon that was vital for fishing and salt production. This decentralized political structure fostered strong local identities and allowed for a degree of genetic differentiation between towns, even as they shared a common heritage. Their settlement on the coast, however, placed them directly in the path of the emerging Atlantic trade. The strategic location of Accra, with its natural harbor, made it exceptionally attractive to European traders. In a remarkably short span of time, 3 rival European powers built forts in close proximity to each other. The English built James Fort, the Dutch built Usher Fort, and the Danes built Christiansborg Castle. This concentration of European presence created an intense and competitive environment for trade, and also for cultural and genetic exchange. The Ga-Dangme people, particularly the Ga-Mashi of Accra, engaged in deep and sustained relationships with the inhabitants of these forts. As with other coastal groups, unions between European men and Ga women were common, leading to the establishment of prominent Euro-African families who played a crucial role in the region's commerce and politics. Surnames like Richter, Hansen, and Bannerman became integral parts of the Ga social fabric. Furthermore, the Ga-Dangme story is uniquely complicated by the arrival of the Tabom people in the 19th century. The Tabom were a group of Afro-Brazilian former slaves who, upon gaining their freedom in Brazil, chartered a ship and returned to Africa, choosing to settle in Accra. They brought with them not only skills in agriculture and architecture, but also a unique genetic heritage forged in the crucible of the Americas, a mix of various African origins with some degree of Portuguese and indigenous American admixture. They were welcomed by the Ga people and integrated into their society, adding yet another fascinating layer to the city's gene pool. The DNA of the Ga-Dangme is therefore a remarkable confluence, a foundational lineage from an Eastern migration, overlaid with centuries of interaction and intermarriage with their Akan neighbors, further enriched by a concentrated and multi-pronged admixture from the English, Dutch, and Danes, and finally complemented by the unique genetic contribution of the returned Afro-Brazilians. This multi-layered history makes the Ga-Dangme a microcosm of the diverse forces that have shaped Ghana's coastal DNA. In the southeastern corner of Ghana, primarily in the region flanking the Volta River, reside the Ewe People, a group with a powerful narrative of migration, a fierce spirit of independence, and a genetic heritage that connects them strongly to lands east of the modern Ghanaian border. Their story is a testament to the idea that political boundaries are often recent impositions on much older patterns of human movement and kinship. The Ewe gene pool is a distinct stream within the Ghanaian mosaic, shaped by a foundational exodus and a subsequent history of decentralized settlement and cross-border connections. Central to Ewe identity is the shared oral tradition of a migration from the east, specifically from the walled city of Notsi in modern-day Togo. According to the tradition, the Ewe people escaped the tyrannical rule of King Agokali by walking backward out of a breach in the city's mud walls, a clever ruse to disguise their tracks. This dramatic escape narrative, known as the Hogbet Sotso, is more than just folklore. It is a unifying charter that binds the numerous Ewe subgroups together and points to a common ancestral origin in the east, likely in the vicinity of the Yoruba city-state of Ketu in present-day Benin. This eastward origin is reflected clearly in their genetics and language. The Ewe language belongs to the B family of languages, which are spoken across Togo, Benin, and southwestern Nigeria, making it distinct from the Kwa languages spoken by their Akan and Ga-Dangme neighbors in Ghana. Genetically, the Ewe share strong affinities with their linguistic cousins across the border, carrying DNA markers that are common among Yoruba and Fon populations. They represent a significant wave of migration that brought a distinct Eastern genetic signature into the Volta Basin. After their exodus from Notsi, the Ewe did not coalesce into a single unified kingdom like the Ashanti. Instead, they settled in a variety of ecological niches, from the coastal lagoons to the fertile inland valleys, and established numerous independent chiefdoms and settlements. This political fragmentation, while sometimes a military weakness, allowed for the development of rich local traditions and a degree of genetic drift between different Ewe communities. It created a pattern of localized genetic variation within the broader framework of their shared ancestral heritage. Their geographical location also placed them at a natural crossroads. They engaged in trade, warfare, and intermarriage with the Akan kingdoms to their west, particularly the Akwamu, and with the Ga-Dangme to the south. This resulted in a degree of gene flow, blending their Eastern heritage with influences from central Ghana. Furthermore, the Volta region experienced a different colonial history from the rest of the Gold Coast. It was first colonized by Germany and later administered by Britain as part of the League of Nations mandate of British Togoland. This brought them into contact with different European groups and created a colonial border that artificially divided the Ewe-speaking people, with a significant population remaining in what is now Togo. This shared cross-border identity persists to this day, with family and genetic ties easily spanning the international frontier. The DNA of the Ghanaian Ewe is thus a story of a distinct founding population from the east, which brought a B-Yoruboid genetic legacy to the Volta region. This foundation was then shaped by a history of decentralized settlement, interaction with neighboring Ghanaian groups, and a unique colonial experience, making them another vital and unique contributor to the rich genetic tapestry of Ghana. In the vast, sun-drenched savanna of northern Ghana, the history and genetic makeup of the Dagomba people are dominated by a single, transformative event: the arrival of a small group of heavily armed, horse-mounted warriors from the northeast around the 15th century. This event, immortalized in the rich oral traditions surrounding the legendary figure Nabi Wa, was not a mass migration but an elite conquest that fundamentally reshaped the region's political, social, and genetic landscape. The resulting Dagomba Kingdom, along with the related states of the Mamprusi and Nanumba, provides a classic example of how a process of elite dominance can leave a clear and lasting signature on a population's DNA. Before the arrival of Nabi Wa and his followers, the region was inhabited by a number of disparate, largely stateless agricultural societies, often referred to collectively as the Tindamba, singular, Tindana, or custodians of the land. These indigenous groups had their own deep history in the region, likely descending from some of the earliest inhabitants of the savanna. The newcomers, who oral traditions suggest came from the Lake Chad region or perhaps from Zamfara in modern Nigeria, possessed a superior military technology: the horse, which was a formidable weapon in the open savanna, and a more centralized model of political organization. They were able to quickly establish dominance over the local populations. The process that followed was not one of extermination but of integration and stratification. The incoming warrior elite, who became known as the Namo, established themselves as the ruling class, while the indigenous population formed the bulk of the commoner class. A complex social contract emerged, where the Namo held political and military power, while the Tindamba retained their spiritual authority as the original custodians of the earth. This social structure had direct and predictable genetic consequences. The conquest was carried out by men, the warriors of Nabi Wa's lineage. They established a strictly patrilineal system of succession for chieftaincy, from the paramount king, the Yana, down to the village level. This meant that their Y-chromosomal DNA, passed directly from father to son, became strongly associated with the ruling class. This specific set of paternal lineages, originating from the small founding group of conquerors, was amplified over the centuries as the ruling class grew and spread its authority. Genetic studies in the region have reflected this history, showing that certain Y-DNA haplogroups are found at a significantly higher frequency among individuals from the ruling lineages compared to the general population. In stark contrast, the mitochondrial DNA, mtDNA, which tracks the maternal line, tells a very different story. The incoming warriors took wives from the local populations they subdued. Therefore, the maternal gene pool of the Dagomba Kingdom is much more diverse and shows a much stronger continuity with the pre-existing indigenous groups. It represents the genetic heritage of the majority population that was incorporated into the new state. This creates a fascinating genetic dichotomy: a relatively homogeneous set of paternal lineages associated with the founding elite, superimposed upon a much older and more diverse maternal genetic foundation. Over centuries of intermarriage, the lines have blurred, but the foundational event has left an unmistakable imprint. The DNA of the Dagomba people is a living document of this historical synthesis, telling a story of conquest, accommodation, and the creation of a new, stratified society from the fusion of two different peoples. It is a powerful illustration of how political power and social structure can directly channel the flow of genes, creating a unique genetic legacy written in the patrilines of kings. In the heart of the dense rainforest of central Ghana, a political and social transformation occurred in the late 17th century that would not only create one of Africa's most powerful pre-colonial empires, but also act as a formidable engine for genetic consolidation. The rise of the Ashanti Empire, a federation of Akan states unified under the leadership of Osei Tutu I and the spiritual authority of the Golden Stool, was a pivotal event that dramatically reshaped the flow of genes in the region. It acted as a powerful crucible, taking a collection of related but distinct Akan groups and forging them into a new, more homogeneous, and formidable cultural and genetic entity. Before the empire's formation, the Akan people were organized into a multitude of competing states and chiefdoms. While they shared a common language, culture, and the all-important matrilineal clan, a bourgeois system, centuries of separate development had likely led to a degree of localized genetic differentiation. The unification of Ashanti changed everything. The empire embarked on a period of rapid and sustained military expansion, conquering and incorporating a vast number of neighboring Akan and non-Akan peoples. This imperial expansion was a powerful mechanism for genetic mixing. The capital, Kumasi, grew into a large, cosmopolitan city, a magnet for people from all corners of the empire and beyond. Traders, artisans, warriors, and captives from dozens of different ethnic groups were brought into the Ashanti heartland. This created an unprecedented level of population movement and intermarriage within a large, centralized state, breaking down previous geographic and ethnic barriers to gene flow and leading to a significant blending of previously distinct gene pools. The structure of the Ashanti state itself facilitated this process. The empire's formidable army was a national institution that brought together men from every province, fostering a shared identity and also serving as a vector for gene flow as soldiers formed relationships and had families in the territories where they were stationed or on their return home. Furthermore, the state's immense wealth, derived from its control over the region's rich gold mines and its strategic position on trade routes, created a stable and prosperous core that encouraged population growth and attracted migrants. This process of consolidation and homogenization occurred in a fascinating interplay with the persistent matrilineal system. While the empire's political and social forces were mixing paternal lineages, Y-DNA, on a grand scale, the bourgeois system continued to meticulously preserve the integrity of the ancient maternal clans. An Ashanti person's primary identity remained tied to their mother's lineage, and the distinct mtDNA signatures of the Oyoko, Adwana, Asona, and other clans were maintained. This created a unique genetic situation where the population was becoming more homogeneous overall, particularly from a paternal perspective, while simultaneously preserving a deep and ancient structure of maternal diversity. The Ashanti Empire, therefore, did not simply erase previous genetic identities, but rather superimposed a new, broader imperial identity upon them. It acted as a great genetic blender for the forest region, creating a more unified Ashanti profile, while the cultural DNA of the matriclan system ensured that the foundational maternal lineages endured. The empire's legacy is thus a dual one. It created a powerful political and cultural identity that defines central Ghana to this day, and in doing so, it fundamentally reshaped the genetic landscape, creating a consolidated Akan gene pool that is a direct product of its imperial history. Along the central coastline of Ghana, the Fante people, a major Akan group, forged a unique destiny shaped by their strategic location, their distinctive political organization, and, above all, their prolonged and intimate relationship with the European powers who came to trade. For nearly 400 years, the Fante lived in the very shadow of the great European trade castles at Cape Coast, Elmina, and Anomabu. This sustained proximity created a zone of intense cultural and genetic interaction that was unparalleled in the interior, leaving a distinct and discernible European imprint on the Fante gene pool. Unlike the centralized, autocratic Ashanti Empire, the Fante were organized into a loose confederacy of independent states. This flexible alliance, often led by the powerful state of Mankasim, allowed them to unite for common defense, particularly against Ashanti expansionism, while preserving their local autonomy. This political structure, combined with their position as the primary middlemen in the Atlantic trade, gave them a unique degree of control and influence in their dealings with the Europeans. They were not conquered subjects but powerful and savvy partners. and the relationships that developed were complex and often mutually beneficial. This partnership extended from the commercial to the personal. From the earliest days of Portuguese contact, a system of long-term relationships between European men, traders, soldiers, and administrators, and local Fante women became institutionalized. Known as casar or calixir, a local corruption of the Portuguese word casar, meaning to marry. These were not casual affairs but strategic alliances that were vital for business and diplomacy. A European trader would form a bond with a woman from an influential local family, gaining access to her kinship network and her knowledge of local trade practices. In return, the woman and her family gained prestige and direct access to European goods and wealth. The children of these unions, the Euro-African Fante, formed a new and influential social class. They were a bridge between worlds, often bearing European surnames, receiving Western education, sometimes even being sent to Europe, and possessing a unique cultural fluency that allowed them to thrive as brokers and leaders in the coastal society. This process, sustained over many generations and involving not just the Portuguese but also the Dutch, English, and Danes, resulted in a steady and significant flow of European Y-chromosomal DNA into the Fante population. While this type of admixture occurred all along the Gold Coast, its concentration and social significance were arguably greatest in the Fante areas. The sheer number and proximity of the European forts in their territory created more opportunities for these interactions. The result is a higher frequency and diversity of European paternal haplogroups, like the Western European R1b and the Scandinavian I1, among the Fante compared to most other Ghanaian ethnic groups. Modern Fante families with surnames like Dawson, Jackson, Brew, Swansea, and Lang are the direct descendants of these historic unions. The Fante DNA, therefore, tells a compelling story of an Akan people whose genetic trajectory was fundamentally altered by their embrace of the Atlantic world. Their foundational Akan heritage was profoundly reshaped by centuries of direct, sustained, and often institutionalized intermarriage with Europeans, creating a unique coastal genetic signature that stands as a living testament to their history as the great brokers of the Gold Coast. While much of Ghana's genetic story is one of addition, of migrations, admixture, and the layering of new peoples upon old, its most traumatic chapter is one of catastrophic subtraction. The Transatlantic Slave Trade, which ravaged the Gold Coast from the 16th to the 19th century, was a demographic and genetic catastrophe on an unimaginable scale. It was a violent, systematic, and prolonged hemorrhage of human beings, representing a massive and forced outflow of the region's very DNA. This brutal commerce did not just steal individuals. It ripped entire lineages out of the West African gene pool, leaving behind deep and permanent scars in the form of population bottlenecks, skewed sex ratios, and a profound loss of genetic diversity that reshaped the biological landscape of the survivors. The Gold Coast was a major epicenter of this trade. Its forts, such as Cape Coast and Elmina, were not just trading posts but dungeons and points of no return, processing hundreds of thousands of captives for the horrific Middle Passage. The demand for enslaved labor in the Americas was insatiable, and it fundamentally reoriented the political and military dynamics of the region. States like the Ashanti Empire and Akwamu rose to prominence in part by becoming formidable slave-raiding powers, waging wars against their neighbors not for territory or tribute in the traditional sense, but for the express purpose of capturing people to sell to the Europeans. This created a terrifying internal dynamic of predator and prey states. The genetic consequences were devastating and complex. For the societies that were systematically raided, groups in the northern savannah and the stateless peoples of the Central Belt were particularly vulnerable. The impact was a severe genetic bottleneck. Their populations were decimated, leading to a drastic reduction in genetic diversity. Entire mitochondrial and Y-chromosomal lineages, representing millennia of unique evolutionary history, were extinguished in the region or were drastically reduced in frequency as their carriers were shipped across the Atlantic. The constant raiding also created a profound sex imbalance. The slavers, both European and African, overwhelmingly preferred young, healthy men for the physically demanding labor on plantations. This meant that far more men than women were forcibly removed from the Gold Coast. The societies left behind faced a severe shortage of men, which had long-term genetic consequences, potentially leading to a reduction in Y chromosomal diversity and forcing changes in social structures and marriage patterns. Conversely, for the powerful slave-raiding states like Ashanti, the genetic impact was different but equally profound. Their populations were not depleted in the same way. In fact, their military success and control over the trade often led to population growth in their core territories. However, their role as captors also shaped their gene pool. They incorporated large numbers of female captives into their societies as wives, concubines, and domestic slaves. This resulted in a massive influx of new mitochondrial DNA from diverse, often non-Akan ethnic groups into the Ashanti maternal gene pool, even as their core paternal lineages remained relatively stable. The DNA of the Ashanti, therefore, bears the complex signature of this history, a record of both their own Akan ancestry and the maternal lineages of the many peoples they conquered and assimilated through the mechanisms of the slave trade. The trade's legacy is a ghost in the Ghanaian genome, a story told not just by the genes that are present, but by the ones that are conspicuously absent, lost forever to the Americas. It created a genetic landscape pockmarked by loss and distorted by violence, an irreversible alteration that stands as the most painful and profound chapter in the making of modern Ghana's DNA. In the intricate tapestry of Ghanaian genetics, woven from threads of ancient migrations, imperial conquests, and coastal trade, there exists a single vibrant strand that traveled across the Atlantic and back again. This is the story of the Tabom People, a small but culturally significant community in Accra whose origins lie not in a Ghanaian village or a Sahelian Empire, but in the tumultuous world of 19th century Brazil. Their return to Africa in 1836 is a remarkable tale of reverse migration, a testament to the resilience of African identity, and unique genetic event that introduced a novel and complex blend of DNA into the GA society of the capital. The Tabom were a group of Afro-Brazilian freed slaves, primarily of Yoruba descent, who had been caught up in the male revolt of 1835 in Salvador, Bahia. This was a major urban slave rebellion organized by African Muslims, and its suppression by the Brazilian authorities was swift and brutal. In the aftermath, facing persecution and fearing re-enslavement, a group of these freed Africans pooled their resources, purchased a ship, the British brig SS Salisbury, and made the audacious decision to return to the continent of their ancestors. They did not know exactly where they were going, only that they were seeking a home in Africa. After a perilous journey, they landed on the coast of the Ga Kingdom, in the heart of what would become Accra. They arrived as strangers in a strange land, speaking Portuguese and practicing trades they had learned in Brazil, such as tailoring, architecture, and irrigation farming. According to Ga oral history, when the locals asked them who they were, they would reply, "Tabom," a common Portuguese phrase meaning, "it's okay," or "I'm fine." The Ga people began to refer to them as the Tabom, and the name stuck. The Ga Mantse, the king of the Ga people, welcomed these newcomers, granting them land in a part of the city that is still known as the Brazil House area. The Tabom quickly integrated into Ga society, their skills making them a valuable asset to the community. They introduced new crops like mangoes and cassava, and their distinctive architectural styles can still be seen in parts of Old Accra. They embraced Ga culture, adopted Ga names alongside their Portuguese ones, like de Souza, de Costa, and Silva, and began to intermarry with their Ga neighbors. This intermarriage initiated a unique genetic infusion. The DNA of the Tabom people was itself a complex mosaic. While predominantly of West African origin, largely Yoruba from modern-day Nigeria and Benin, their ancestors had spent generations in Brazil, a vast genetic melting pot. During this time, it is highly probable that their gene pool had acquired some degree of admixture from other populations in Brazil, primarily from European, Portuguese, men and, to a lesser extent, from the indigenous Amerindian peoples of South America. Therefore, when the Tabom integrated into the Ga community, they introduced not only a concentrated dose of Yoruba lineages that had been absent from Accra, but also trace amounts of European and potentially even Native American DNA that had been carried back across the Atlantic. This was a genetic signature unlike any other in Ghana, forged in the crucible of the New World diaspora and then reintroduced into the African mainland. Today, the descendants of the Tabom are fully integrated into the fabric of Accra, yet they retain a proud sense of their unique history. Their story is a powerful counter-narrative to the one-way tragedy of the slave trade. It demonstrates that the Atlantic, while primarily a chasm of loss, could also, on rare occasions, be a bridge for return. Genetically, they represent a fascinating footnote, a small but complex loop in the flow of the African diaspora, adding a distinct, transcontinental layer to the already cosmopolitan DNA of Ghana's capital city. Far from the forested kingdoms of the south and the bustling coastal forts, the vast savanna and Sahelian fringe of northern Ghana constitutes a world apart, a region whose genetic landscape is arguably the most complex and deeply layered in the entire country. For millennia, this region has been a zone of convergence, a crossroads where different linguistic, cultural, and genetic streams have met, mingled, and clashed. It lacks the relative homogeneity of the Akan heartland or the specific coastal admixture of the Fante. Instead, it is a true mosaic, a patchwork of diverse lineages reflecting a history shaped by ancient autochthonous populations, waves of migration from every direction, and the rise and fall of powerful cavalry states. The foundational genetic layer of the north belongs to the Gur-speaking peoples, such as the Grusi, the Sisola, and the Konkamba. These groups are widely considered to be among the most ancient inhabitants of the region, the descendants of populations who have farmed this land for thousands of years. Their DNA represents a deep-rooted savanna heritage, adapted to the specific ecological challenges of the area. They were traditionally organized into stateless, decentralized societies, governed by councils of elders and spiritual leaders known as Tindamba, earth priests, who held custody over the land. This ancient stratum was profoundly disrupted from the 15th century onwards by the arrival of migrating groups from the northeast, most notably the ancestors of the Mole-Dagbani peoples, the Dagomba, Mamprusi, and Nanumba. As detailed in their oral traditions, these were politically organized, horse-mounted warriors who established centralized kingdoms, superimposing their rule upon the indigenous Gur populations. This created a stratified society and a distinct genetic pattern of elite dominance, where a specific set of paternal lineages, Y-DNA, associated with the conquering class became widespread among the nobility, while the maternal gene pool, mtDNA, remained largely rooted in the more numerous indigenous groups. But the Mole-Dagbani were not the only newcomers. The Northern Territories were also a major corridor for Monde-speaking groups from the west, particularly the Daila and Ligbai, who were instrumental in the trans-Saharan trade. They established merchant communities and brought with them genetic lineages more typical of the old Mali and Songhai empires. From the north and east came Hausa traders, establishing the Zongo communities that became hubs of commerce and Islamic learning, adding another layer of genetic influence from what is now northern Nigeria. Furthermore, the region has long been a zone of interaction with nomadic and semi-nomadic pastoralist groups, most notably the Fulani. For centuries, Fulani herders have moved through the savanna with their cattle, engaging in trade and sometimes settling and intermarrying with local agriculturalist communities, introducing specific Sahelian genetic markers, including those for lactase persistence, into the local gene pool. This history of multi-directional migration and interaction has made the North a region of incredible genetic diversity. It is not uncommon to find neighboring villages where the predominant paternal and maternal lineages are. While much of Ghana's genetic story is one of addition, of migrations, admixture, and the layering of new peoples upon old, its most traumatic chapter is one of catastrophic subtraction. The Transatlantic Slave Trade, which ravaged the Gold Coast from the 16th to the 19th century, was a demographic and genetic catastrophe on an unimaginable scale. It was a violent, systematic, and prolonged hemorrhage of human beings, representing a massive and forced outflow of the region's very DNA. This brutal commerce did not just steal individuals. It ripped entire lineages out of the West African gene pool, leaving behind deep and permanent scars in the form of population bottlenecks, skewed sex ratios, and a profound loss of genetic diversity that reshaped the biological landscape of the survivors. The Gold Coast was a major epicenter of this trade. Its forts, such as Cape Coast and Elmina, were not just trading posts but dungeons and points of no return, processing hundreds of thousands of captives for the horrific Middle Passage. The demand for enslaved labor in the Americas was insatiable, and it fundamentally reoriented the political and military dynamics of the region. States like the Ashanti Empire and Akwamu rose to prominence in part by becoming formidable slave-raiding powers, waging wars against their neighbors not for territory or tribute in the traditional sense, but for the express purpose of capturing people to sell to the Europeans. This created a terrifying internal dynamic of predator and prey states. The genetic consequences were devastating and complex. For the societies that were systematically raided, groups in the northern savannah and the stateless peoples of the Central Belt were particularly vulnerable. The impact was a severe genetic bottleneck. Their populations were decimated, leading to a drastic reduction in genetic diversity. Entire mitochondrial and Y-chromosomal lineages, representing millennia of unique evolutionary history, were extinguished in the region or were drastically reduced in frequency as their carriers were shipped across the Atlantic. The constant raiding also created a profound sex imbalance. The slavers, both European and African, overwhelmingly preferred young, healthy men for the physically demanding labor on plantations. This meant that far more men than women were forcibly removed from the Gold Coast. The societies left behind faced a severe shortage of men, which had long-term genetic consequences, potentially leading to a reduction in Y-chromosomal diversity and forcing changes in social structures and marriage patterns. Conversely, for the powerful slave-raiding states like Ashanti, the genetic impact was different but equally profound. Their populations were not depleted in the same way. In fact, their military success and control over the trade often led to population growth in their core territories. However, their role as captors also shaped their gene pool. They incorporated large numbers of female captives into their societies as wives, concubines, and domestic slaves. This resulted in a massive influx of new mitochondrial DNA from diverse, often non-Akan ethnic groups into the Ashanti maternal gene pool, even as their core paternal lineages remained relatively stable. The DNA of the Ashanti, therefore, bears the complex signature of this history, a record of both their own Akan ancestry and the maternal lineages of the many peoples they conquered and assimilated through the mechanisms of the slave trade. The trade's legacy is a ghost in the Ghanaian genome, a story told not just by the genes that are present, but by the ones that are conspicuously absent, lost forever to the Americas. It created a genetic landscape pockmarked by loss and distorted by violence, an irreversible alteration that stands as the most painful and profound chapter in the making of modern Ghana's DNA. In the intricate tapestry of Ghanaian genetics, woven from threads of ancient migrations, imperial conquests, and coastal trade, there exists a single vibrant strand that traveled across the Atlantic and back again. This is the story of the Tabom people, a small but culturally significant community in Accra whose origins lie not in a Ghanaian village or a Sahelian empire, but in the tumultuous world of 19th century Brazil. Their return to Africa in 1836 is a remarkable tale of reverse migration, a testament to the resilience of African identity, and unique genetic event that introduced a novel and complex blend of DNA into the Ga society of the capital. The Tabom were a group of Afro-Brazilian freed slaves, primarily of Yoruba descent, who had been caught up in the male revolt of 1835 in Salvador, Bahia. This was a major urban slave rebellion organized by African Muslims, and its suppression by the Brazilian authorities was swift and brutal. In the aftermath, facing persecution and fearing re-enslavement, a group of these freed Africans pooled their resources, purchased a ship, the British brig SS Salisbury, and made the audacious decision to return to the continent of their ancestors. They did not know exactly where they were going, only that they were seeking a home in Africa. After a perilous journey, they landed on the coast of the Ga Kingdom, in the heart of what would become Accra. They arrived as strangers in a strange land, speaking Portuguese and practicing trades they had learned in Brazil, such as tailoring, architecture, and irrigation farming. According to Ga oral history, when the locals asked them who they were, they would reply, "Tabom," a common Portuguese phrase meaning, "it's okay," or "I'm fine." The Ga people began to refer to them as the Tabom, and the name stuck. The Ga Mandse, the king of the Ga people, welcomed these newcomers, granting them land in a part of the city that is still known as the Brazil House area. The Tabom quickly integrated into Ga society, their skills making them a valuable asset to the community. They introduced new crops like mangoes and cassava, and their distinctive architectural styles can still be seen in parts of Old Accra. They embraced Ga culture, adopted Ga names alongside their Portuguese ones, like de Souza, de Costa, and Silva, and began to intermarry with their Ga neighbors. This intermarriage initiated a unique genetic infusion. The DNA of the Tabom people was itself a complex mosaic. While predominantly of West African origin, largely Yoruba from modern-day Nigeria and Benin, their ancestors had spent generations in Brazil, a vast genetic melting pot. During this time, it is highly probable that their gene pool had acquired some degree of admixture from other populations in Brazil, primarily from European, Portuguese, men and, to a lesser extent, from the indigenous Amerindian peoples of South America. Therefore, when the Tabom integrated into the Ga community, they introduced not only a concentrated dose of Yoruba lineages that had been absent from Accra, but also trace amounts of European and potentially even Native American DNA that had been carried back across the Atlantic. This was a genetic signature unlike any other in Ghana, forged in the crucible of the New World diaspora and then reintroduced into the African mainland. Today, the descendants of the Tabom are fully integrated into the fabric of Accra, yet they retain a proud sense of their unique history. Their story is a powerful counter-narrative to the one-way tragedy of the slave trade. It demonstrates that the Atlantic, while primarily a chasm of loss, could also, on rare occasions, be a bridge for return. Genetically, they represent a fascinating footnote, a small but complex loop in the flow of the African diaspora, adding a distinct, transcontinental layer to the already cosmopolitan DNA of Ghana's capital city. Far from the forested kingdoms of the south and the bustling coastal forts, the vast savanna and Sahelian fringe of northern Ghana constitutes a world apart, a region whose genetic landscape is arguably the most complex and deeply layered in the entire country. For millennia, this region has been a zone of convergence, a crossroads where different linguistic, cultural, and genetic streams have met, mingled, and clashed. It lacks the relative homogeneity of the Akan heartland or the specific coastal admixture of the Fante. Instead, it is a true mosaic, a patchwork of diverse lineages reflecting a history shaped by ancient autochthonous populations, waves of migration from every direction, and the rise and fall of powerful cavalry states. The foundational genetic layer of the north belongs to the Gur-speaking peoples, such as the Grusi, the Sisola, and the Konkamba. These groups are widely considered to be among the most ancient inhabitants of the region, the descendants of populations who have farmed this land for thousands of years. Their DNA represents a deep-rooted savanna heritage, adapted to the specific ecological challenges of the area. They were traditionally organized into stateless, decentralized societies, governed by councils of elders and spiritual leaders known as Tindamba, earth priests, who held custody over the land. This ancient stratum was profoundly disrupted from the 15th century onwards by the arrival of migrating groups from the northeast, most notably the ancestors of the Mole-Dagbani peoples, the Dagomba, Mamprusi, and Nanumba. As detailed in their oral traditions, these were politically organized, horse-mounted warriors who established centralized kingdoms, superimposing their rule upon the indigenous Gur populations. This created a stratified society and a distinct genetic pattern of elite dominance, where a specific set of paternal lineages, Y-DNA, associated with the conquering class became widespread among the nobility, while the maternal gene pool, mtDNA, remained largely rooted in the more numerous indigenous groups. But the Mole-Dagbani were not the only newcomers. The Northern Territories were also a major corridor for Monde-speaking groups from the west, particularly the Daila and Ligbai, who were instrumental in the trans-Saharan trade. They established merchant communities and brought with them genetic lineages more typical of the old Mali and Songhai empires. From the north and east came Hausa traders, establishing the Zongo communities that became hubs of commerce and Islamic learning, adding another layer of genetic influence from what is now northern Nigeria. Furthermore, the region has long been a zone of interaction with nomadic and semi-nomadic pastoralist groups, most notably the Fulani. For centuries, Fulani herders have moved through the savannah with their cattle, engaging in trade and sometimes settling and intermarrying with local agriculturalist communities, introducing specific Sahelian genetic markers, including those for lactase persistence, into the local gene pool. This history of multi-directional migration and interaction has made the North a region of incredible genetic diversity. It is not uncommon to find neighboring villages where the predominant paternal and maternal lineages are quite distinct, reflecting different local histories of settlement and assimilation. The North is where the genetic influences of the Guinea Coast forest meet those of the Sahel and Sahara. It is a buffer, a bridge, and a battleground, and its DNA reflects this complex role. It is a mosaic of ancient Gur heritage, overlaid with the patrolines of Mole-Dagbani kings, interwoven with the threads of Monde and Hausa traders, and flecked with the genetic signature of Fulani pastoralists, making it a microcosm of the great historical and demographic currents that have swept across West Africa for centuries. While the grand narratives of Ghana's genetic history are dominated by large-scale African migrations and the long shadow of European contact, a smaller, more recent, and economically significant stream of DNA arrived in the late 19th and early 20th centuries from the Eastern Mediterranean. The arrival of merchants and traders from what was then the Ottoman province of Syria, encompassing modern-day Lebanon and Syria, introduced a distinct Levantine genetic signature into the Ghanaian social fabric. Though small in number, their remarkable success in commerce and their subsequent integration into Ghanaian society created a new and influential thread in the country's complex genetic tapestry, demonstrating that even minor demographic movements can have a disproportionate social and genetic impact. These early migrants, often referred to collectively as Lebanese, though many were Syrian Christians, were driven from their homeland by a combination of economic hardship, political instability under Ottoman rule, and a desire for new opportunities. They did not arrive as part of a state-sponsored colonial project but as independent, enterprising individuals and families. Initially, many intended to travel to the Americas, but through various quirks of fate and shipping routes, they found themselves in the bustling port cities of West Africa, including Accra, Sekondi, and later, the burgeoning commercial hub of Kumasi. They arrived with little capital but a strong entrepreneurial spirit and tight-knit kinship networks that were crucial to their success. They quickly identified and filled a critical niche in the colonial economy, acting as intermediaries between the large European trading firms and the small-scale African producers and consumers in the hinterland. They established shops and trading posts, dealing in textiles, hardware, and imported provisions, and became major players in the cocoa trade, extending credit to farmers and arranging for the transport of beans to the coast. Their success was built on a willingness to live and work in remote areas that European agents often avoided, and on building personal relationships with their Ghanaian customers and suppliers. As their businesses prospered, these merchant families became a permanent and visible part of Ghanaian urban society. Unlike the transient European colonial administrators, the Lebanese and Syrians put down roots. They built homes, established community organizations, and became integral to the economic life of the country. This permanence inevitably led to social and genetic integration. While first-generation migrants often sought spouses from their home villages in the Levant, subsequent generations increasingly intermarried with the local Ghanaian population. These unions were particularly common between Lebanese or Syrian men and Ghanaian women, often from prominent local families. This process introduced a new set of paternal and maternal lineages into the Ghanaian gene pool, primarily from the Middle East. DNA haplogroups such as J1 and J2, common Y-DNA markers in the Levant, and various West Eurasian mitochondrial DNA lineages began to appear in families that were, for all intents and purposes, Ghanaian. The children of these unions grew up in a unique bicultural environment, fluent in local languages like Twi or Ga as well as Arabic, and comfortable in both Ghanaian and Levantine social settings. Surnames like Fokri, Captain, Trabulsi, and Hage became well known in Ghanaian commerce and, eventually, in politics and the professions. The genetic contribution of this Levantine migration is, in percentage terms, very small across the entire population. However, it is concentrated in specific urban centers and within particular influential families, making it a significant element of the country's elite social structure. It stands as a powerful example of how modern global migrations, driven by commerce, can add new layers of complexity to a nation's genetic identity, weaving the DNA of the Middle East into the rich and ancient fabric of West Africa. The establishment of the British Gold Coast colony in the late 19th century did not involve mass European settlement, and therefore did not result in the kind of large-scale genetic replacement seen in other parts of the world. However, the colonial project, driven by its own economic and administrative imperatives, acted as a powerful, albeit often indirect, force for reshaping the genetic landscape of Ghana. British rule did not primarily alter the gene pool through direct admixture, but rather by fundamentally re-engineering the colony's infrastructure and economy, which in turn catalyzed unprecedented internal migrations, broke down old ethnic barriers, and created new centers of genetic mixing. The colonial impact was most profound in its creation of a modern, centralized infrastructure designed to facilitate the extraction of resources. The construction of a railway network was perhaps the single most important factor. Lines were driven from the coastal ports of Sekondi and Accra deep into the interior, their primary purpose being to transport cocoa and gold from the Ashanti and eastern regions. These railway lines acted as powerful new arteries for human movement. For the first time, it became possible for large numbers of people to travel quickly and cheaply across long distances that had once required weeks of arduous trekking. The railway towns that sprang up along these routes, places like Takwa, Obwesai, and Sawam, became magnets for people seeking work. The gold mines of the western region and the burgeoning cocoa farms of the Brong-Ahafo and Ashanti regions created a massive demand for labor. This demand was met by a wave of migration, primarily from the less economically developed northern territories. Young men from Dagomba, Frofra, and Walla communities traveled south in their thousands to work in the mines and on the farms. This was a demographic shift on a scale never seen before. These migrant laborers, who often stayed for years or settled permanently, lived and worked alongside the local Akan populations. This created entirely new social environments where inter-ethnic contact, and consequently intermarriage, became commonplace. A Frofra man working in an Obwesai gold mine might marry a local Ashanti woman, creating a family with a novel combination of northern and southern genetic lineages. The colonial administration also established new urban centers that became powerful melting pots. Accra was solidified as the capital, and its civil service, port, and commercial enterprises drew in educated and ambitious individuals from every single ethnic group in the colony. Similarly, Kumasi, the old Ashanti capital, was transformed into a major administrative and commercial hub for the interior, attracting a diverse population. In these cities, ethnic identity, while still important, was often secondary to professional or social status. People from different backgrounds lived as neighbors, worked in the same offices, and their children attended the same schools. This urban environment fostered a new, more national consciousness and provided a fertile ground for the erosion of traditional ethnic endogamy. The colonial project, therefore, acted as a great, unintentional genetic blender. By creating a unified economic and political space, and by building the infrastructure that allowed people to move within it, it broke down the relative isolation of many of Ghana's ethnic groups. It initiated a process of large-scale internal gene flow from north to south and from rural to urban areas, a process that would accelerate dramatically after independence. The genetic patterns of modern Ghana, particularly the complex admixture found in its major cities, are a direct consequence of these colonial-era transformations that set the population in motion. The Volta River system, with its vast basin and sprawling man-made lake, is more than just a geographical feature of Ghana. It is a historical and genetic corridor that has functioned for millennia as a crucible of human diversity. This region, a complex transition zone of riverine, savanna, and forest environments, has been a natural channel for migration, a meeting point for disparate peoples, and a refuge for fragmented communities. Its unique geography has fostered a level of ethnic and linguistic diversity that is remarkable even by Ghanaian standards, resulting in a genetic landscape of incredible complexity, characterized by the interdigitation of major West African language groups and the preservation of ancient, relict populations. The basin's role as a genetic crossroads predates all modern ethnic identities. Its fertile riverbanks and accessible waterways have made it an attractive area for settlement since prehistoric times. It was a key area for the dispersal of early agriculturalists and ironworkers. However, its most defining characteristic is its role as a meeting point for three of West Africa's major linguistic and, by extension, genetic groupings. From the west came the Kwa-speaking peoples, most notably various Akan groups who expanded towards the river, establishing states like Akwamu, which at its height controlled a vast stretch of the lower Volta. From the east, the Bi-speaking Ewe people migrated into the basin, settling the lands east of the river in their flight from Notsi. And from the north, Gur-speaking peoples have long inhabited the upper reaches of the basin. The Volta River itself has often acted not as a barrier, but as a highway, facilitating trade, communication, and intermarriage between these groups. This has created a broad zone of genetic clines, where the characteristic markers of Akan, Ewe, and Gur populations blend and overlap. But the story of the Volta Basin is not just about the convergence of large populations. It is also about the preservation of unique and ancient ones. The rugged hills and less accessible areas of the basin, particularly in the region now known as the Oti region, became a refuge for numerous smaller ethnic groups. These are the Ghana-Togo mountain languages communities, often referred to as remnant or relict populations. Groups like the Guan, the Avatime, the Santrakofi, and the Akpafu, among many others, are believed to represent some of the earliest inhabitants of the region, predating the arrival of the larger Akan and Ewe migrations. They were pushed into these more defensible highland areas by the expansion of their more powerful neighbors. This historical process of displacement and isolation has had profound genetic consequences. For centuries, these smaller groups have had relatively little gene flow from the outside, allowing them to preserve unique and ancient genetic lineages that may have been lost or diluted in the larger surrounding populations. They are, in effect, genetic islands, providing invaluable windows into the deeper ancestral history of West Africa. The Gwan people are a particularly fascinating case. They are considered to be among the very first Kwa speakers to settle in Ghana, and their fragmented communities are found scattered from the coast to the northern regions, often along the Volta. Their DNA tells a story of a once widespread population that was later overlaid and assimilated by the Akan expansion, with pockets of their original genetic signature surviving in places like the Afram Plains and the Akwapim Hills. The construction of the Akosombo Dam in the 1960s and the creation of Lake Volta, the world's largest artificial reservoir, was a modern cataclysm that dramatically reshaped this ancient landscape. It displaced some 80,000 people from over 700 villages, forcing diverse communities to resettle together in new townships. This massive, state-engineered resettlement project initiated a new and intense phase of genetic mixing in the region, bringing together groups that had previously lived in relative isolation and accelerating the creation of a new, complex Volta identity. The achievement of independence in 1957 was not just a political milestone for Ghana. It was a profound social and demographic catalyst that unleashed and accelerated population movements on an unprecedented scale. The nationalist vision of Kwame Nkrumah, centered on creating a unified nation-state and driving rapid industrialization, fundamentally altered the incentives for where and how Ghanaians lived. This era ushered in a massive wave of internal migration, primarily from rural areas to a few rapidly expanding urban centers, transforming cities like Accra, Kumasi, and Sekondi Tokorodi into dynamic genetic melting pots. This process, more than any other in the modern era, is responsible for the complex, pan-ethnic admixture that characterizes urban Ghanaian DNA today. Under colonial rule, movement had been significant but was largely channeled towards specific economic zones like the mines and cocoa farms. After independence, the drivers of migration diversified and intensified. The new government invested heavily in education, healthcare, and infrastructure, but this development was heavily concentrated in the major southern cities. Accra, as the national capital, became the epicenter of political power, administration, and higher education. Kumasi continued its role as the commercial heart of the country, while the twin city of Sekondi Tokorodi was developed as a major industrial and port hub. These cities became beacons of opportunity, promising jobs, education, and a modern lifestyle that was unattainable in most rural villages. This promise triggered a human torrent. Millions of Ghanaians left their ancestral hometowns and flocked to the cities. This was not just the north to south labor migration of the colonial era. It was a nationwide phenomenon. Ewe fishermen, Fante traders, Ashanti farmers, Dagomba civil servants, and Kwahu entrepreneurs all converged on the same urban spaces. They came seeking work not just in factories and ports, but in the burgeoning civil service, in schools and hospitals, and in the vast informal economy that grew to serve the needs of the expanding urban population. In these new urban environments, traditional social structures and rules governing marriage began to break down. While first-generation migrants might retain strong ties to their home villages and seek spouses from their own ethnic group, this became less practical and less important for their children, who grew up in multi-ethnic neighborhoods and attended national schools. A young person born and raised in a suburb of Accra like Adabroka or Nima would have friends and classmates from every corner of the country. Their social world was not defined by the narrow confines of their ancestral village, but by the cosmopolitan reality of the city. As a result, inter-ethnic marriage rates, which had been relatively low in the pre-colonial and colonial eras, began to soar. An Ashanti man might marry a Ga woman. A new woman might marry a Frofra man. These unions, once rare, became increasingly common, driven by shared education, professional life, and simple proximity. Each of these marriages was a small act of genetic fusion, combining paternal and maternal lineages that had, for centuries, been largely geographically and culturally separate. This process has created a distinct, urban Ghanaian genetic profile. While an individual from a rural village might have a genome that overwhelmingly reflects their specific ethnic heritage, a person whose grandparents all migrated to Accra is likely to carry a far more complex mix of DNA from across the country. Their genetic signature is a testament to the nation-building project of the post-independence era, a biological reflection of the motto, "One Nation, One People, One Destiny." These cities are now the primary crucibles where a new, truly national Ghanaian genetic identity is being forged, one marriage and one generation at a time. Beyond the grand narratives of migration and conquest, the uniqueness of Ghanaian DNA is also written at the molecular level, in the subtle variations of the genetic code that have allowed its people to survive and thrive in a challenging environment. Perhaps the most dramatic and well-understood example of this is the story of the sickle cell trait, a powerful illustration of natural selection in action. The high prevalence of the gene responsible for sickle cell disease in Ghana and across West Africa is not a flaw or a defect, but a remarkable and costly evolutionary adaptation to a relentless and deadly selective pressure: the malaria parasite, Plasmodium falciparum. This single genetic mutation tells a profound story of a biological trade-off, a genetic compromise forged over thousands of years in a deadly embrace between a human genome and one of its most ancient microscopic foes. Malaria has been a scourge in West Africa for millennia. The hot, humid climate provides a perfect breeding ground for the Anopheles mosquito, the vector that transmits the parasite. Plasmodium falciparum causes the most severe form of the disease, which, if left untreated, is often fatal, particularly in young children who have not yet developed immunity. This devastating mortality rate has exerted an immense selective pressure on the human populations living in the region. Any individual who happened to be born with a genetic mutation that offered even a slight survival advantage against malaria would be more likely to live to reproductive age and pass that advantageous gene on to their children. Over many generations, such a gene would become increasingly common in the population. The sickle cell trait is the result of such a mutation. It is a single-point mutation in the beta-globin gene, which codes for a subunit of hemoglobin, the protein in red blood cells that carries oxygen. Individuals who inherit two copies of this mutated gene, one from each parent, develop sickle cell anemia, a severe and often life-shortening disease where red blood cells become rigid and crescent-shaped, blocking blood flow and causing pain and organ damage. However, individuals who inherit only one copy of the mutated gene, and one normal copy, are said to have the sickle cell trait. They are generally healthy and asymptomatic, but crucially, they are highly resistant to malaria. The presence of the abnormal hemoglobin in their red blood cells makes it difficult for the malaria parasite to replicate and survive. This is known as heterozygote advantage. In a malarial environment, this creates a powerful balancing selection. Individuals with two normal copies of the gene are highly susceptible to malaria. Individuals with two mutated copies suffer from sickle cell disease. But the heterozygotes, those with one of each, are protected from both severe malaria and sickle cell disease. They have the highest fitness, and therefore the sickle cell gene is maintained at a high frequency in the population, despite the devastating consequences for those who inherit two copies. In Ghana, the frequency of the sickle cell trait, HBS, is among the highest in the world, with up to 30% of the population in some areas carrying at least one copy of the gene. This is not a random occurrence. It is a direct biological testament to the historical and ongoing threat of malaria. The distribution of the gene is not uniform, often correlating with areas where malaria transmission has been historically most intense. The sickle cell story is a stark reminder that the human genome is not a static blueprint, but a dynamic, living document that records the history of our ancestors' struggles for survival. The DNA of many Ghanaians carries this molecular scar, a silent but eloquent signature of an ancient and ongoing evolutionary battle fought in the bloodstream of every generation. The ability of an adult human to digest lactose, the primary sugar in milk, is a fascinating and relatively recent evolutionary adaptation. For most of human history, and for the majority of the world's population today, the gene that produces the lactase enzyme, which breaks down lactose, is switched off after weaning. The persistence of this gene's activity into adulthood, known as lactase persistence, evolved independently in a few populations around the world that practiced pastoralism and relied heavily on the milk of domesticated animals as a source of nutrition. In West Africa, the story of lactase persistence is intricately linked to the history of one particular group, the Fulani people. The presence of the lactase persistence allele in various Ghanaian populations, particularly in the north, is a distinct genetic echo of centuries of interaction, trade, and intermarriage with these nomadic and semi-nomadic cattle herders. The Fulani are a people of uncertain deep origins, but they are known for their long history as the preeminent pastoralists of the Sahel and West Africa. For centuries, they have migrated across the vast savanna belt, from Senegal to Cameroon, with their distinctive long-horned zebu cattle. Their entire culture, economy, and diet are centered around their herds. Milk and dairy products are not just supplementary foods for the Fulani. They are staples, providing a rich source of protein, fat, and calories in an often harsh and unpredictable environment. This intense reliance on dairy created a powerful selective pressure. Any individual with a genetic mutation that allowed them to continue producing the lactase enzyme into adulthood would have had a significant nutritional advantage. They would have been healthier, better nourished, and more likely to survive and have more children than their lactose-intolerant peers. Over many generations, the allele for lactase persistence became nearly ubiquitous among Fulani populations. The specific genetic mutation responsible for lactase persistence in West Africa is different from the one found in European populations, indicating a clear case of convergent evolution, where different populations independently evolved the same trait in response to similar environmental pressures. For centuries, Fulani pastoralists have moved through and settled in the northern regions of modern-day Ghana. Their relationship with the settled agriculturalist communities, such as the Ga-Dangme, Mamprusi, and various Gur-speaking groups, has been complex, involving trade, competition for resources, and, crucially, social and genetic exchange. Fulani herders would trade their milk, butter, and livestock for grains and other goods produced by the farmers. Over time, these economic relationships often led to more permanent ties. Fulani communities settled, and intermarriage between Fulani men and local women, or vice versa, occurred. With every such union, there was a chance for the Fulani-associated lactase persistence allele to be passed into the gene pool of the local agriculturalist population. This has created a distinct genetic pattern observable today. The frequency of the lactase persistence allele in Ghana is not uniform. It is highest in the northern populations that have had the most prolonged and intensive contact with the Fulani, and it becomes progressively rarer as one moves south into the forest belt, where cattle herding is difficult due to the tsetse fly and where dairy has never been a significant part of the traditional diet. Therefore, the presence of this specific genetic trait in a Ghanaian individual can be seen as a faint but clear genetic signature of this historical interaction. It is a molecular marker that tells a story of cultural and economic symbiosis between nomadic pastoralists and settled farmers, a story of how the DNA of a mobile, cattle-herding people became woven into the genetic fabric of the savanna communities of northern Ghana. For millennia, long before the advent of modern pharmaceuticals, the peoples of Ghana have relied on the rich biodiversity of their environment for their health care. The practice of traditional herbal medicine is a deeply ingrained and highly sophisticated system of knowledge, passed down through generations, involving the use of hundreds of different plant species to treat a vast array of ailments. This long and intimate relationship between people and plants has not been a one-way street. Just as humans have shaped the landscape to cultivate medicinal plants, it is increasingly plausible that the plants themselves, through the potent biochemical compounds they contain, have exerted a subtle but significant selective pressure on the human genome. The unique genetic profile of Ghanaians may therefore include specific variants in metabolic genes that have co-evolved with the long-term consumption of local herbal remedies, representing a form of pharmacogenetics, written by tradition rather than by prescription. Every plant produces a range of secondary metabolites, compounds like alkaloids, terpenes, and flavonoids, that are not essential for its basic survival but are used for defense against herbivores and pathogens. These are the very compounds that often have medicinal effects in humans. When a person ingests a herbal remedy, their body must process these foreign compounds, primarily using a family of enzymes in the liver known as the cytochrome P450 (CYP) system. These enzymes are responsible for metabolizing a huge variety of substances, from dietary toxins to modern drugs. The genes that code for these CYP enzymes are highly variable across human populations. Different individuals have different versions, or alleles, of these genes, which can make their enzymes faster or slower at processing specific compounds. This genetic variation is the basis of the modern field of pharmacogenomics, which explains why different people respond differently to the same dose of a drug. It is conceivable that in a population that has relied for thousands of years on a specific set of medicinal plants, natural selection would have favored genetic variants that improved the ability to process these local remedies effectively. For example, imagine a widely used herbal treatment for a common and serious illness. If one version of a CYP enzyme metabolizes the plant's active compound into its therapeutic form very efficiently, individuals with that genetic variant would be more likely to recover from the illness and survive to pass on their genes. Conversely, if another variant metabolizes the compound so slowly that it becomes toxic, or so quickly that it has no effect, individuals with those variants would be at a disadvantage. Over many generations, the favorable metabolic allele would become more common in the population. While research in this specific area is still emerging, the principle is well established. For instance, the genetic diversity in genes related to bitter taste perception is thought to have been shaped by the need to detect and avoid poisonous plants. It is a logical extension to assume that the genes involved in metabolizing plant compounds after ingestion have also been under selective pressure. The traditional pharmacopoeia of Ghana is particularly rich, with plants like Cryptolepis sanguinolenta, used for malaria, Graphonia simplicifolia, a source of 5-HTP, and Voacanga africana, containing ibogaine-like alkaloids, being used for centuries. A systematic study of the genetic variation in the metabolic pathways associated with the compounds in these specific plants could reveal a unique Ghanaian genetic signature. It would be a testament to a long history of medical innovation and a deep, co-evolutionary relationship with the local flora, showing how culture, in this case, the culture of traditional healing, can leave a subtle but enduring imprint on the very DNA of its practitioners. Deep within the human genome lies a family of genes so crucial to our survival, so exquisitely sensitive to the pressures of our environment, that they serve as a living molecular chronicle of our ancestors' struggles with disease. This is the human leukocyte antigen, HLA system, a group of genes on chromosome 6 that code for the proteins responsible for distinguishing the body's own cells from foreign invaders. In the unique and pathologically dense environment of Ghana, this genetic region has been under relentless and multifaceted selective pressure for millennia, resulting in a repertoire of HLA alleles that is extraordinarily diverse and distinct. This HLA profile is not merely a biological curiosity. It is a finely tuned immunological weapon, forged in the crucible of a uniquely challenging disease landscape and representing one of the most profound signatures of adaptation in the Ghanaian genome. The primary function of the HLA system is to present fragments of proteins, known as antigens, to the immune system's T cells. If the antigen is from one of the body's own proteins, the T cell recognizes it as self and leaves it alone. If, however, the antigen is from a virus, bacterium, or parasite that has invaded a cell, the T cell recognizes it as non-self and launches a powerful immune attack. The incredible diversity of pathogens in the world means that no single HLA molecule can bind to and present all possible foreign antigens. Therefore, the strength of a population's collective immune defense lies in the diversity of its HLA genes. The more varied the HLA alleles within a population, the wider the range of pathogens that can be effectively recognized and combated. For the people of Ghana, this has been a matter of life and death. The region is a hyperendemic zone for a staggering array of infectious agents, from the ancient scourge of Plasmodium falciparum malaria to viruses like yellow fever and Lassa fever, bacteria causing tuberculosis and meningitis, and a host of parasitic worms. This constant, multi-pronged assault has acted as a powerful evolutionary sieve. Individuals whose HLA types were poorly suited to recognizing the locally prevalent pathogens were less likely to survive and reproduce, while those with HLA alleles that conferred resistance were more likely to pass on their advantageous genes. This process, repeated over countless generations, has sculpted a unique HLA landscape in Ghana. It is characterized by immense polymorphism, the existence of many different alleles for each HLA gene, and the presence of specific alleles that are rare or absent in other parts of the world but are common in West Africa because they offer protection against a local threat. The complex history of migration and admixture within Ghana has further amplified this diversity. The ancient Kintampo people, the southward migrating Proto-Akan, the eastward moving Ga-Dangme and Ewe, and the cavalry states of the north did not just bring new cultures and languages. They brought their distinct HLA profiles, shaped by the different disease environments of their ancestral homelands. The subsequent centuries of intermarriage and population mixing within the geographical space of modern Ghana created a vast combinatorial library of these alleles, generating new HLA haplotypes, combinations of alleles inherited together, and providing the population with an even broader and more robust immunological toolkit. This diversity is a double-edged sword, as the HLA system is also deeply implicated in autoimmunity. Certain alleles that are excellent at fighting off a particular virus may also predispose an individual to conditions like type 1 diabetes or rheumatoid arthritis. The specific balance of HLA types found in Ghana is therefore a unique signature of this evolutionary trade-off, a record of compromises made in the long co-evolutionary war between the Ghanaian genome and its microscopic adversaries. Studying these alleles offers a way to perform a kind of molecular archaeology, revealing the history of past epidemics and the diseases that have been the most powerful agents of selection, making the HLA system a profound and living testament to the resilience and adaptation of the Ghanaian people. In the vibrant, syncopated, and utterly unique sound of highlife music, one can hear an audible echo of the very same historical processes of migration, admixture, and synthesis that forged the Ghanaian genome. Highlife is more than just a musical genre. It is a cultural expression of a genetic reality, a sonic metaphor for the creation of a modern Ghanaian identity from a multitude of diverse ancestral streams. Born in the bustling cosmopolitan port cities of the Gold Coast in the late 19th and early 20th centuries, its very DNA is a fusion of the local and the foreign, the ancient and the modern. The story of highlife begins with its foundational genetic stratum, the indigenous musical traditions of the coastal peoples. The intricate interlocking guitar patterns of Akan palm wine music, the call and response vocal traditions, and the complex polyrhythms of Ga drumming provided the essential melodic and rhythmic bedrock of the new sound. This represents the ancient, autochthonous layer of the Ghanaian gene pool, the deep-rooted heritage of the people who had inhabited the land for millennia. However, this indigenous music did not exist in a vacuum. The coastal cities were the primary points of contact with the outside world, the very same spaces where European DNA was first introduced into the Fante and Ga populations. Into this musical environment came a wave of foreign influences, each acting like a new stream of gene flow. The brass bands of the colonial regiments introduced new instruments, trumpets, trombones, saxophones, and new harmonic concepts based on Western scales and chord progressions. This was the European admixture, a powerful and transformative element that was adopted, adapted, and reinterpreted by local musicians. At the same time, sailors and stevedores from across the Atlantic world brought with them the sounds of the African diaspora, Caribbean calypso and Trinidadian mento, with their own complex histories of African and European fusion, introduced new syncopations and a distinctive lilting feel. This represents a form of reverse migration, a reintroduction of African cultural genetic material that had been transformed in the crucible of the Americas, much like the return of the Tabom people from Brazil. From the Americas also came the influence of jazz and swing, brought via records and radio, which added a new layer of improvisational sophistication and cool to the burgeoning genre. The genius of the early highlife pioneers was their ability to act as master synthesizers, to take these disparate musical elements and weave them into a seamless and coherent whole that was entirely new, yet unmistakably Ghanaian. The name highlife itself speaks to this synthesis, referring to the high-class bowls where these new dance bands played, events that were themselves melting pots of the new African elite and European officials. As the music matured and spread from the coast into the interior, it continued to evolve and incorporate new elements, mirroring the post-independence migrations that blended the country's diverse peoples in the growing cities. The guitar band highlife that flourished in the Ashanti heartland had a different flavor from the sophisticated dance band style of Accra, reflecting regional cultural and genetic variations. The music absorbed influences from the Ewe, the Ga-Dangme, and other groups, becoming a truly national art form. Highlife, therefore, is a perfect allegory for the Ghanaian genome. Its foundational structure is indigenous West African, but it has been profoundly enriched and transformed by centuries of interaction with Europe, the Americas, and the wider African diaspora. It is a product of a society comfortable with fusion, a culture adept at taking foreign elements and making them its own. Listening to the sweet, intricate interplay of a highlife band is to hear the story of Ghana itself, a complex, beautiful, and resilient synthesis born from a history of convergence at a global crossroads. The DNA of the Ghanaian people is one of the world's great, unread historical archives. It is a living library, its molecular pages filled with the epic stories of ancient migrations, the rise and fall of empires, the devastating impact of disease, and the remarkable power of human adaptation. For centuries, this history could only be glimpsed through the lenses of archaeology, linguistics, and oral tradition. But today, the tools of modern genomics offer the potential to read this archive directly, to translate its genetic code into a high-definition narrative of the past. The prospect of a comprehensive Ghana Genome Project, a large-scale, systematic effort to sequence and analyze the genomes of people from across the nation's diverse ethnic landscape, represents a monumental scientific undertaking, promising not only to revolutionize our understanding of Ghanaian history, but also to unlock secrets of profound importance for human health and our shared human story. Such a project would be founded on the recognition that Ghana is a place of extraordinary and globally significant human genetic diversity. As the cradle of humanity, Africa harbors more genetic variation than the rest of the world combined. And Ghana, as a historical crossroads of major population movements, is a microcosm of this continental richness. A Ghanaian genome project would therefore aim to create a detailed genetic map of the nation, sequencing the full genomes of thousands of individuals from a carefully selected cohort representing the full spectrum of the country's ethnic and linguistic groups, from the Akan of the forest to the Dagomba of the savannah, from the Ewe of the Volta to the isolated Guan communities, and from the coastal Fante to the Grusi of the far north. The revelations from such a dataset would be transformative. By comparing the genetic sequences of different groups, researchers could reconstruct the history of population movements with unprecedented precision. They could test the validity of oral traditions, identifying the genetic signatures of the Akan migration from the north or the Ewe exodus from Notsi. They could date the divergence of different ethnic groups, map the precise routes of ancient trade networks by tracing the flow of specific lineages, and quantify the genetic impact of historical events like the rise of the Ashanti Empire or the Transatlantic Slave Trade. Beyond history, the project would hold immense medical promise. By correlating genetic variants with health data, scientists could identify the specific genes that have allowed Ghanaians to adapt to their challenging disease environment. This could lead to the discovery of new genetic factors for resistance to malaria, Lassa fever, or tuberculosis, potentially paving the way for new vaccines or therapies that could benefit people worldwide. It could also shed light on why people of West African descent have some of the world's highest rates of certain non-communicable diseases, such as hypertension, type 2 diabetes, and specific cancers, by uncovering the genetic predispositions and understanding how they interact with modern lifestyles. This knowledge would be critical for developing preventative healthcare strategies tailored to the specific biological realities of the Ghanaian population. Crucially, for such a project to succeed ethically and sustainably, it must be led by Ghanaian scientists and institutions. It would represent a powerful act of scientific sovereignty, ensuring that the biological heritage of the nation is studied by its own people, for its own benefit. It would be a monumental step towards decolonizing the world of genomics, which has for too long focused overwhelmingly on populations of European descent. By meticulously reading the stories encoded in its own DNA, Ghana would not only illuminate its own unique past but would also provide the entire world with a richer, more complete understanding of the magnificent tapestry of human diversity and history. For centuries, the story of gene flow in Ghana was primarily one of inward movement, of migrations into its territory and the arrival of newcomers on its shores. The major exception, the catastrophic outflow of the Transatlantic Slave Trade, was a story of violent rupture and genetic loss. However, in the latter half of the 20th century and into the 21st, a new chapter in the nation's genetic story began to be written, one characterized by a massive, voluntary, and global dispersal. The modern Ghanaian diaspora represents a new and profound outflow of the country's unique genetic heritage, a peaceful and purposeful seeding of its complex DNA into the wider human gene pool, fundamentally altering the genetic landscapes of nations on every continent. This great migration began as a trickle in the early years of independence, as students and diplomats traveled abroad, but it swelled into a flood during the periods of economic and political instability in the 1970s and 1980s. Unlike the forced diaspora of the past, this was a movement of choice, driven by the universal human desires for education, economic opportunity, and security. Ghanaians from every ethnic group and every walk of life, doctors, engineers, academics, traders, and laborers, made their way to new homes, with major communities establishing themselves in the United Kingdom, the United States, Germany, Canada, Italy, and the Netherlands. This diaspora carried with it the entirety of Ghana's complex genetic history. Each individual who settled in London, New York, or Hamburg was a living archive, their genome containing the layered stories of the Kintampo farmers, the Akan matriclans, the Sahelian traders, the Ga fishermen, the Ewe migrants, and the northern kingdoms. They carried the genetic signatures of adaptation to malaria, the markers of lactase persistence from Fulani contact, and the faint but discernible traces of European admixture from the coastal forts. This rich and uniquely West African genetic legacy was now being directly introduced into the populations of Europe and North America on a significant scale for the first time. As these diaspora communities have matured, they have become powerful new centers of genetic mixing. While first-generation migrants often maintained strong ties to home and married other Ghanaians, their children and grandchildren, born and raised in new cultural contexts, have increasingly formed partnerships and had families with people from outside the Ghanaian community. This has initiated a new and fascinating phase of global admixture. A child born in South London to a mother of Ashanti descent and a father of English-Irish heritage represents a novel fusion of West African and Western European lineages. A family in Toronto with a Fante father and a mother of Chinese-Canadian descent creates a genetic bridge between Africa and Asia in a way that was historically impossible. In the Bronx, the children of a Ga father and a Puerto Rican mother embody a new synthesis of African and Afro-Caribbean Amerindian European histories. This process is fundamentally different from the unidirectional gene flow of the colonial era. It is a multi-directional exchange that is enriching the genetic diversity of the host nations while simultaneously creating new, transnational identities. The modern Ghanaian diaspora is not a story of genetic subtraction, but of multiplication and diversification. It represents the Ghanaian genome breaking its geographic confines and participating fully in the ongoing creation of a global, cosmopolitan human gene pool. The unique combination of alleles, forged over millennia in the crucible of West Africa, is now being shared with the world, ensuring that the epic and complex story written in the DNA of Ghana will become a permanent and integral part of the future story of humanity itself. The story of Ghana's DNA is, in its essence, a story of convergence. It is a chronicle of how disparate streams of humanity, with their own unique histories and genetic inheritances, have flowed into a single geographical basin, mingling and merging over centuries to create a new, vibrant, and indivisible whole. This profound genetic legacy, characterized by deep diversity and intricate interconnectedness, is not merely a biological fact. It is the fundamental underpinning of the modern Ghanaian nation-state and a powerful resource for its future. The nation's celebrated stability and social cohesion in a region often marked by ethnic strife can be traced, in part, to this shared and complex biological heritage. While Ghana is composed of over 70 distinct ethnic groups, its history has not been one of rigid, impermeable genetic silos. Instead, it has been a continuous process of fusion. The arteries of trade, the upheavals of state formation, the shared experience of colonialism, and the unifying project of nation-building have all acted as powerful engines of admixture, weaving the genetic threads of Akan, Ewe, Ga, Ga-Dangme, and Ga into a single, complex national fabric. Understanding this shared legacy is crucial for the future of Ghanaian identity. In an era where ethnic chauvinism can be a potent and destructive force, genomics offers a powerful counter-narrative. It provides scientific validation for the idea that there is no pure Ashanti, pure Ewe, or pure Ga-Dangme. Rather, it reveals that all Ghanaians are, to varying degrees, a blend of the same ancestral components, sharing deeper connections than superficial cultural or linguistic differences might suggest. The ability for an individual to discover, through a DNA test, that their own genome contains markers from groups they once considered other, has the potential to be a profoundly unifying experience, dissolving prejudice and fostering a more inclusive sense of national belonging. This process of genetic fusion is not a relic of the past. It is accelerating in the present and will define the future. The great urban centers of Accra and Kumasi are the modern crucibles where a new Ghanaian identity is being forged in the most literal sense. Here, in the schools, offices, and markets, young people from every corner of the country meet, form relationships, and build families, creating a generation of children whose very DNA embodies the nation's motto of one people. These urban Ghanaians, with their multi-ethnic parentage, are the living vanguard of a future where ethnic identity becomes just one layer of a more complex and cosmopolitan sense of self. As genetic technologies become more accessible, the relationship between Ghanaians and their own ancestral history will become more personal and more profound. This will raise new questions about identity. What does it mean to be Akan when one's genome also tells a story of Sahelian and Ewe ancestors? How does a family in the north reconcile their patrilineal identity with the discovery of deep maternal lineages from the southern forests? Navigating these questions will be a central challenge and opportunity for 21st century Ghanaian society. Ultimately, Ghana's genetic legacy is its greatest strength. It is a testament to a history of adaptation, resilience, and an extraordinary capacity for assimilation and synthesis. It proves that diversity is not a source of weakness but the very foundation of a dynamic and robust society. The future of Ghanaian national identity lies in embracing this complexity, in celebrating the multitude of stories encoded in the DNA of its people, and in recognizing that the ongoing vibrant process of genetic and cultural mixing is the truest expression of the nation's unique and enduring spirit. This narrative explores the deep genetic currents that have shaped a nation. From ancient migrations to modern diasporas, Ghana's DNA tells a remarkable story of resilience, adaptation, and fusion. What did you find most surprising? And how can this story be improved for future viewers? Your feedback is welcome.